Okay. Well, good morning to those of you in the US. Good afternoon to those of you in Europe, and a good late evening to those of you in Asia. It's a pleasure to present to you now. I'm Edward Dennis. I was asked to talk about fatty acids and eicosanoids. And I am going to do this by emphasizing their structure and function. Let me say at the beginning that I'm going to try to pitch this as I would for first year or second year graduate students who are not familiar with all of the background and details of lipids, as I was asked. And so apologies to the experts in the audience, but I'm happy to address more in-depth questions during the question and answer period and the question and answer period that follows. So I'm going to talk about fatty acids and eicosanoids in 5 parts. First, while I recognize that Valerie O'Donnell introduced lipid maps and lipid categories, and Matt Conroy talked much about the classification and categories, I want to begin by introducing lipid maps, lipidomics, and lipid categories from the unique perspective of fatty acids and eicosanoids. And then I want to dwell in detail on fatty acids as components of both simple and complex lipids. Then we'll turn to the formation of oxygenated polyunsaturated fatty acids, the so-called PUFAs, and these are eicosanoids and related oxylipins. Then I want to give a brief example of eicosanoids and data from lipidomics as an illustration of their role and how one looks at those. And I want to mention a potential conflict of interest as a company called Liponexus works on fatty liver disease. And finally, I want to introduce the phospholipases because they have a subsite in their catalytic site that is very specific for precise fatty acids. And those specificities can detect whether there's an extra double bond or an extra couple of carbons in a fatty acid chain to emphasize the very important structural basis of fatty acids and their recognition in biological settings. So with that, let me introduce lipid maps once more, following up on Valerie. And lipid map stands for lipid metabolites and pathways strategy. And it really has its origin in the genomics revolution, which is practiced, in fact, as transcriptomics. But in hindsight, genomics was really quite simple because all one has to do in genomics or transcriptomics is recognize which of the 4 bases is at a particular position in the polymer backbone. And as we now know, there are only about 25,000 coding genes in the human genome. Proteomics is more challenging because there are 20 amino acid side chains and pre and post translational modification. And with that, there are about 30,000 unique proteins in the databases. But if we turn to metabolomics, which includes all the molecules of a cell, nucleic acids, amino acids, sugars, and fats, there's over 100,000 unique metabolites. And in the databases, there's about 48,000. Now, when we just focus on lipidomics or all the fats, as this training course is focused, there's still over 100,000 unique lipid molecules. And in the lipid maps database, as of this morning, there were 45,684 unique lipids. Now you'll quickly, if you do the math from metabolomics to lipidomics, you'll see that the vast majority of metabolites that we know with unique structures are lipids, and only a relatively small number of nucleic acids, amino acids, and sugars. Now, with that, if you look at human plasma, and there was a question to Al Merrill a little bit ago about sphingolipids in human plasma. If you look at human plasma, 3/4 of the metabolites are lipids, and only 1/4 are comprised of the other 3 categories of biological materials: nucleic acids, amino acids, and sugars. If we look at just the lipids, and Matt introduced you to the categories of lipids, of which lipid maps established 8 categories, but only 6 of those are involved actively in human and mammalian metabolism. Those are shown here. And of these 6 categories in human plasma, about half the metabolites are sterols, as Bill Griffith spoke about. The other 5 categories comprise the other half of the lipids in human plasma, as listed here, roughly in proportion to the numbers of them. Now, the smallest little slice of the pie is fatty acyls, and that includes fatty acids and eicosanoids. And that little slice of the pie is what I have been asked to address today. Now, just to note for a minute that when lipid maps first took on the challenge of determining the lipid molecular species in the human plasma, it used a sample of human plasma, which was a very large pooled sample developed by the US National Institutes of Standards. And this was a pooled sample of plasma, like a liter of blood, from 100 individuals. They were half female and half male. They did have a narrow age range of 40 to 50, and they represented the ethnic population of the US at the time the sample was actually collected, which is some 20 years ago now, and it's changed since then. But that's what this pooled sample is. Lipid Maps used it, and many other lipidomics initiatives through the succeeding decade have used this same sample for standardization. Now, when Lipid Maps determined the human plasma lipidome, it identified some 600 molecular species. When I say molecular species, I'm using Matt's definition of lipid species, which is with full structural knowledge and stereochemical detail, as that was the standard Lipid Maps used from the beginning. And shown here on this sort of abbreviated metabolic chart in orange is the number of lipid molecular species for each of the categories. If you look at the center of the slide, you'll see that fatty acids, there were 31 molecular species of free fatty acids, but many of them were oxygenated to form icosanoids. The icosanoids, together with the fatty acids, comprise 2 of the main classes of fatty acyls. Now, fatty acids are synthesized from acetyl-CoA, polymerized through fatty acid synthase to make, from acetyl-CoA to make fatty acids. These are the so-called simple lipids, fatty acids and their derivatives like icosanoids. But there are many more complex lipids than there are free fatty acids, because fatty acids are esterified to various backbones: glycerolipids, glyceryl phospholipids, and sphingolipids, as indicated across the top. Now, there's another category of complex lipids. Those are synthesized from acetyl-CoA through isopentyl pyrophosphate at the bottom of the slide. And these categories are called isoprenoids. And those backbones give rise to prenyls and to the sterol backbone, as Bill Griffiths spoke about. I want to point out, however, that even the sterol backbone can be esterified with fatty acids. And most of our cholesterol in our body, which is perhaps the most abundant of lipids, especially in plasma, is cholesterol ester with the fatty acid on it. So this is a quick overview to the place of fatty acids as simple fatty acyls, along with eicosanoids, and complex lipids. So let's now focus on the second topic, which is fatty acids. And here is the flow chart for how we synthesize fatty acids, starting with the product of fatty acid synthase, which is palmitic acid. We can elongate it and we can desaturate it to put a single double bond at the 9 position to make oleate. However, we humans and other mammals cannot put any additional double bonds further down the fatty acid chain than position 9. Only plants can do that. And so the simple fatty acid linoleate with 2 double bonds is an essential fatty acid for us humans. Now, if we start with linoleate, the essential fatty acid, we can add double bonds before position 9, and that leads by the pathway on the right to arachidonic acid, the most important polyunsaturated fatty acid in metabolism. It has 20 carbons and 4 double bonds. But there's another set of important fatty acids, the so-called omega-3 fatty acids, present in fish oil. Now, they're derived from linoleate also, but only plants can put a double bond at position 15 to make alpha linoleate and other polyunsaturated fatty acids of the omega-3 variety. Now, I don't like looking at flow charts, and I'm sure you don't like memorizing charts of how fatty acids are made. So I prefer to look at the cabbage and the rabbit being eaten by the rabbit, in which the cabbage represents the plant kingdom, which makes linoleate and linolenate the 2 essential fatty acids. Rabbits, plants, and humans eat it, and they convert it by a number of enzymatic steps over to the right to arachidonic acid from linoleate. And on a little bit to the left, they convert the omega-3 fatty acid precursor essential fatty acid to EPA and DHA, eicosapentaenoic acid and DHA. I will leave it at. So these are the key polyunsaturated fatty acids that are not only fatty acids, but are precursors for the eicosanoids and related oxylipins. Before I turn to those, I just want to talk a bit about its nutrition and presence. And as everyone is probably aware, fish oils contain a relatively high concentration of omega-3 fatty acids, principally DHA and EPA, can be as much as 1/3 of the fatty acid in fish oils. But other oils contain very little. Linseed oil has a lot, but no one wants to eat it. And soybean oil contains a small amount, but mostly omega-6. The other popular nutrition-based fatty acids have no omega-3 fatty acids and have an abundance of omega-6 fatty acids. And many of them have very little of that as well, and mostly saturated fatty acids. So with that, I want to turn to my third topic, which is eicosanoids and related oxylipids. But I want to start with this slide of inflammation, because inflammation is the one disease that was known by the Romans and Greeks some 2,000 years ago. And they gave names to these characteristics of inflammation. I won't give the Greek names, but just the English names here: heat, redness, swelling, pain, and loss of function. These characterize inflammation. Inflammation is the one disease that the U.S. National Institutes of Health does not have a separate institute for. They have cancer, heart, diabetes, and every other disease, but inflammation. And that is because it is so pervasive as diseases go. Now, how do we talk about inflammation from the standpoint of lipids and lipid metabolism? And the key to inflammation is one of its major components in the lower left-hand corner of this slide. Can you see my arrow pointing? I'm not sure if you can or not. Can somebody say yes or no?
Yeah.
Okay. So prostaglandins are a key icosanoid, and these are hallmark of inflammatory diseases. Now, let me talk about how one generates prostaglandins and go back to the top of the slide. There are a large variety of membrane receptor events on virtually all cells and tissues of the body that, when activate a key enzyme in lipid metabolism called phospholipase A2. This enzyme specifically hydrolyzes the fatty acid on the middle or SN2 position of membrane phospholipids. And when the fatty acid that is on that middle position happens to be an arachidonyl fatty acid, then free arachidonic acid is liberated. Free arachidonic acid is acted upon by the enzyme cyclooxygenase to make prostaglandins. So let me pause for a second and ask if anyone on this call has ever taken an aspirin or any other non-steroidal anti-inflammatory drug. Say yes or raise your hands if you did.
Yes.
Okay.
Yes.
I'm going to assume that's a wide answer of yes. Now, that's the problem with Zoom calls. You can't see everybody's faces. But let me just say that the sole mode of action of aspirin or any other NSAID is to inhibit the cyclooxygenase and stop prostaglandin synthesis. So anything you might take an aspirin for, a headache, a wound, arthritis, menstrual cramps, and the list goes on, is because of specific prostaglandins causing those syndromes under the general rubric of inflammation. Now, there are other inflammatory mediators, the lipoxygenases, which is a whole category of enzymes, also produce a category of inflammatory mediators called leukotrienes. They're not inhibited by aspirin or NSAIDs, and they tend to be dominant in allergy and asthma in the lung. We won't, in the interest of time, go into more details than that. So let me turn now one more step in detail to the pathway I just described. And this is a schematic picture of a macrophage in which encounters a bacteria like an E. coli or another gram-negative bacteria, which carries lipopolysaccharide or LPS on its surface. That lipopolysaccharide or E. coli, when binding to a surface membrane dissolving receptor called TOL4, leads to the activation of one specific subform of phospholipase A2 called CPLA2. It also induces through NF-kappa B and gene transcription the synthesis of COX-2, one specific form of the cyclooxygenase. Together, PLA2 and COX-2 generate numerous icosanoids. Now, there's another kind of receptor, a purinergic receptor, one of which is a P2X7, it's called, which is activated by ATP. And when ATP binds to the P2X7 dimeric receptor, calcium influx occurs. Calcium is another way of activating CPLA2. It also is a specific requirement for the activation of lipoxygenase enzymes, and that leads to numerous icosanoids as well. Well, let's look at it at another depth of schemes. And if one goes to the middle of this next figure and sees the word phospholipid and the word PLA2, imagine in this scheme a particular subform called CPLA2, leading to, which is specific for releasing arachidonic acid, leading to free arachidonic acid in a cell. That arachidonic acid can be acted upon in the upper left by cyclooxygenases. There are two main ones, COX-2 and COX-1, and these lead to various primary prostaglandins, abbreviated here, which lead to secondary prostaglandins that are listed here. And all these prostaglandins have specific G-coupled protein receptors they activate, which are indicated in green, or they activate various PPARs, which cause transcriptional regulation indicated in orange. Now, this is just one small part of the biosynthesis of icosanoids and related oxylipins, because the arachidonic acid can be used by the so-called 5-lipoxygenase to make leukotrienes and other products that activate a different set of G-coupled protein receptors or PPARs. There are indeed other lipoxygenases, 8-15, 12-15, 12, 12R, a whole number of other lipoxygenases that make various products that also some can activate PPARs and other G-coupled protein receptors like the TRP receptors. And arachidonic acid can be acted upon by P450s to make EETs, which are then converted to hetras and diols, and those can activate various PPARs. In our lipidomics analysis in our lab, which I'll talk about tomorrow, we analyzed some 200 products of arachidonic acid and other PUFAs, particularly the omega-3, EPA, and DHA, for which some of these enzymes can act on as well and make unique products. I want to digress for a second just to mention aspirin, since I dwelled on it as an example, because it is such an important enzyme and inhibitor of the cyclooxygenase, and it's such a simple mechanism. It turns out that the cyclooxygenase enzymes, and there are only two forms of it, have a serine in their active site. It has nothing to do with mechanism of action of the cyclooxygenase or catalytic activity. There just happens to be a spare serine in the catalytic site. And in the catalytic site, it was discovered 100 years ago by the Bayer Corporation in Germany that salicylic acid was a good analgesic and anti-inflammatory, and that it binds in the active site of the cyclooxygenase as a competitive inhibitor. But a smart chemist discovered that if you made acetylated salicylic acid, and that this acetylated form of aspirin would be catalytically acetylate that serine, liberating salicylate, that it was a much stronger inhibitor of inflammation. And it kills the enzyme. It is a permanent covalent inhibitor, and that's why aspirin is perhaps the most widely used drug in the world and indicates every disease that is inflammatory. So now let me go back to the scheme I showed on the slide before the last one. This is the same scheme as I showed 2 slides ago, except it emphasizes drugs and inhibitors. Here's the phospholipid PLA2 making arachidonic acid. In orange are all the drugs and pharmaceutical agents in the world that are used today to block various steps in the icosanoid pathway. And I want to draw your attention to some future developments where people are trying to now develop mimetics of lipoxins, resolvins, and protectins, which are icosanoid-like products that are related oxylipins that are believed to be anti-inflammatory or pro-resolving. So with that, I want to summarize this section by emphasizing there are only 2 COX genes, COX-1 and COX-2. The number of genes in our genome is relatively small. They code, each one codes for a protein, but there's post and pre-translational modification. So there are more proteins than there are genes. But when you go to metabolites, there's a giant expansion because of the number of fatty acids. There are 30 or 40 normal fatty acids that are very abundant in our metabolism, and many of these proteins can work on all of them. So you get an expanding number of metabolites. And I illustrated only with icosanoids why there are so many more icosanoids than there are proteins that act on them or genes. And this is true for the whole lipidome and why there are so many lipid metabolites. With that, I want to just conclude finally this section, because Lipid Maps a decade ago, when it determined the human plasma lipidome, also determined the macrophage lipidome as a typical cell. These were activated macrophages like the scheme I showed you before. And this is a metabolic chart similar to the one for the human plasmidome. And in orange are the number of individual molecular species of each. And again, almost 600 individual molecular species, stereochemically correct and in detail, were determined by Lipid Maps over a decade ago as an early example of cellular lipidomics. So with that, I want to now turn to a little interlude about the importance in a broader way of lipids in disease. And I've chosen to illustrate that with this schematic picture of metabolic syndrome, the pervasive disease of our times, because metabolic syndrome is portrayed as obesity, as insulin resistance and diabetes, and many cardiovascular diseases listed on the right. The latest manifestation of metabolic syndrome is fatty liver disease, and that is technically termed non-alcoholic fatty liver disease. Classically, fatty liver was due to alcoholism. Today, it's due much more to obesity and other factors, and it's called non-alcoholic fatty liver disease, NAFLD. And I introduced this because this is a growing epidemic where about a third of the US population now has fatty liver. And of those, about a third have non-alcoholic steatohepatitis, abbreviated NASH. And there's a progression from fatty liver to NASH to fibrosis and cirrhosis of the liver, and eventually liver cancer, and is soon becoming the major reason for liver transplants in the US, and all this applies worldwide as well. Now, I only introduce this because I want to illustrate an example of the power of lipidomics related to fatty acids and icosanoids. Shown here is just a small pilot study on 10 on plasmas for plasma lipidomics from individuals, 10 individuals with fatty liver disease symbolized by NAFL, and 10 with the advanced form called NASH. And this is the amount of arachidonic acid in their plasma at the center top, which you can see is increased in those with fatty liver and those with NASH. Now, if we look at some of the metabolites of arachidonic acid, shown here to the left is the COX pathway. Down below are the lipoxygenase pathway, and to the right is the P450 pathway, much like in my earlier schemes. I just want to draw your attention to one metabolite, which is called DHK-PGD2. And in this metabolite, those with fatty liver are not very much increased over those of controls, but those with the advanced form of NASH has an increase in this metabolite in their plasma as a diagnostic for this advanced form called NASH. And one can see some other metabolites, the P450 products, which are enhanced specifically in NASH over just fatty liver. Now, these are the metabolites of arachidonic acid. On this next slide are shown metabolites of other fatty acids. Linoleate and linolenic acid are two essential fatty acids, as well as EPA and DHA on the right. And I just want to draw your attention to an example of EPA metabolite 12-HEAP, in which again, there's more of this metabolite in plasma of patients with NASH than in just fatty liver. Or if one looks at the very bottom, a metabolite of DHA called 1920 di-HDPA, there's more in NASH than NAFLD. This is the power of lipidomics to look in plasma or in tissues at changes. And the area under the curve for discriminating between NASH and fatty liver for these two metabolites shown here on the left, or a combination of the two of them, becomes close to 100% for a distinction as to whether one has only fatty liver, which in and of itself is not particularly harmful, versus the advanced stage called NASH. So with that, I want to turn to my final topic, which is a introduction of phospholipases and some specific illustration of the power of stereochemistry and structure of proteins detecting fatty acids. So phospholipase A2 is a superfamily of enzymes. Some of them are degradative, in which phospholipase A2s particularly are enzymes that catalyze the hydrolysis of a fatty acid on the SN2 position of membrane phospholipids. This is shown in the stereochemically correct form that Matt Conroy introduced earlier today. And when the enzymes are just degradative, they produce as products lysophospholipids, and free fatty acids is illustrated. Others are biosynthetic when they're coupled with an acyltransferase to add a fatty acid to the SN2 position of the lysophospholipid product, usually polyunsaturated PUFAs. Others are signaling enzymes when their products are converted by other enzymes to ligands that activate G-coupled protein receptors like LPA and PGE2. Now, there are many phospholipase A2s, and this slide is trying to illustrate some of them. A key one, and the one we're going to focus on today and have already, is called CPLA2. It resides on the Golgi. It's a cytosolic enzyme, and it's specific for releasing arachidonic acid and in instituting inflammatory reactions. Others, like SPLA2, are secreted and have a preference for saturated fatty acids. IPLA2 is cytosolic and prefers linoleic acid at the SN2 position, and lipoprotein-associated PLA2s, which are in our plasma and associated with lipoproteins like LDL, have a great specificity for short-chain or oxidized fatty acids on the 2 position. So these different phospholipase A2s can tell what the structure is of the underlying fatty acid that they're going to cleave if they're activated to carry out hydrolysis. Now, this is the result of a lipidomics experiment. I'm not going to describe the lipidomics, but in which pure cytosolic or CPLA2 is allowed to react with a phospholipid in a mixed mice cell with a non-ionic detergent, and to compare the lipidomics determined activity towards palmitoyl arachidonyl phosphatidic acid, or with the same fatty acids, phosphatidylcholine, phosphatidylethanolamine, phosphatidylglycerol, and phosphatidylserine. Sorry, I know that the introduction to phospholipids will not occur till tomorrow morning by Yunkin Aoki. So I'm preluding that a little bit, but 5 fatty acids. However, when these 5 phospholipids are mixed in a mixed mice cell, as on the bottom, and the enzyme phospholipase A2 is allowed to react with them, the activity toward each of the phospholipids is exactly the same. And what this experiment shows is the specificity of this phospholipase A2 is not for the polar group on a phospholipid, but it works equally on all polar groups when one has an equimolar mixture in a membrane or a mice cell. In contrast, this enzyme in the upper left has a great specificity for the fatty acid at the 2 position, in which it's 20:4, which is arachidonic acid. What I've been speaking about all morning is the best fatty acid in the 2 position for this enzyme. And another enzyme, group 6, IPLA2, doesn't like arachidonic acid. It likes linoleic acid the best. So here are 2 enzymes. One prefers arachidonic 24:4. One prefers linoleic, which is 18:2. And on the top, the fatty acid in the SN1 position is palmitic, or in the bottom, it's stearic, which are saturated fatty acids, which dominate the 1 and 2 position. Now, in order to understand this specificity, this is some work that was carried out in our lab, but is pertinent to the question that we're addressing. We take these phospholipases like CPLA2 we mix them with phospholipid vesicles. We let them associate, and then we dilute them in D2O and look at the exchange rate of deuterium with the amide backbone of the phospholipase to determine what part of the phospholipase enzyme interacts with the membrane. And from that, kinds of experiments, we've developed a model for how this enzyme works. This is a water-soluble enzyme, phospholipase A2. It associates with a membrane, and the membrane causes a conformational change in the enzyme to activate it and expose its active site to binding a phospholipid. And it can pull one phospholipid out of the membrane up into its active site. And think about a phospholipid with a polar group, the circle, and two fatty acid chains. At the very top, as I'm pointing to it, is the binding site for the SN2 or leaving fatty acid chain. And down here is for the SN1 chain. Catalysis occurs, the products diffuse back in the membrane, and the cycle repeats itself. Now, this is the result of those experiments. This is a molecular dynamics simulation in which the catalytic domain of the enzyme, which is shown at the top, in which a single phospholipid substrate is bound in the catalytic site of the enzyme in black. And the enzyme active site is on the one side of a membrane bilayer for which there's a slice shown in purple of the other membrane phospholipids. And it is allowed to carry out molecular dynamics to optimize the binding mode of the phospholipid in the active site of the enzyme. And you can see where my pointer is the SN2 fatty acid chain, which is arachidonic acid in this phospholipid. And the arachidonic acid has a very specific binding site in which the arachidonic acid is a sort of curved structure, because this is how it binds in that catalytic site. Now, in this next simulation, we've pulled this phospholipid out of the active site, and it's in the membrane, and the simulation shows what happens when the enzyme has docked on a membrane and is sucking up a single phospholipid into the active site. So you can see where the active site is and how a single substrate is pulled up into its active site to carry out hydrolysis. And that is the location. Now, on the next slide is shown a different enzyme, the IPLA2, which I already mentioned as preferring linoleic acid, but it's shown here pulling up the same phospholipid as on the previous slide, in which arachidonic acid is in that middle or SN2 position and where it's pulled to. Once it's pulled up in the active site and it's allowed to occupy its ideal position, the active site opens up upon causes a conformation change. And this defines the substrate site of the arachidonic acid in this enzyme. In this enzyme, arachidonic acid looks very different in its structure than in CPLA2 because the binding subsite for the SN2 fatty acid has a very different conformation. And I want to illustrate that in this slide. On the left is CPLA2. This is a phospholipid containing palmitic acid and arachidonic acid and choline as the polar group. And this is how arachidonic acid binds in the catalytic site. In IPLA2, arachidonic acid binds in a very different way in this very specific site. However, on the left is how arachidonic acid normally looks conformationally in X-rays. On the right is a very abnormal conformation. And that's because the IPLA2 shown here is really meant to bind linoleic acid. And here's how it binds linoleic acid in a perfect way in the catalytic site. Linoleic acid is only 18 carbons and 2 double bonds. The whole point I'm making is that CPLA2 is made to bind arachidonic, and IPLA2 is made to bind linoleic, though it will bind arachidonic poorly. And so these enzymes specify which fatty acid, and that's why it's so important whether a fatty acid is 20 carbons or 18 carbons long, or has 2 double bonds or 4 double bonds. So to conclude, the scheme I showed a few minutes ago of a water-soluble phospholipase associating with the membrane causes a conformational change, opening up the active site to bind a single phospholipid with very specific subsites for the SN2 leaving fatty acid, and incidentally for the SN1 fatty acid. Each of the enzymes is different, and there's some small differences for polar groups, but very little, to cleave it to make its products to go back in the membrane and recycle is how these enzymes work, taking advantage of the structural and stereochemical differences of the fatty acid chains. So with that, I want to conclude, and I want to thank my colleagues and co-workers in my lab who contributed to many of the slides and details that I provided today. Highlighted in yellow are the particular individuals who've contributed to the specific data that I showed today. So let me now open it up for questions. I also wanted to note that I have currently a postdoctoral position open for somebody to do cellular lipidomics of eicosanoids, phospholipids, and single lipids. And if anybody is interested or has further questions that aren't answered today, please email me as shown on this slide. With that, I will stop and take questions.